Hello, welcome to 6.5 and 6.6. We are talking about organelles used in energy transfer and the cytoskeleton. So if we're starting to talk about organelles that are used in energy transfer, we do have to start with the endosymbiont theory, which is created by Dr. Lynn Margulies, which talks about the formation of mitochondria in chloroplast. Let's talk quickly about what each of these do. So chloroplasts take light energy from the sun. They're found in plants, and they convert that light energy into a more usable form called chemical energy stored in the bonds of glucose. And so that is the job of chloroplasts. Mitochondria then take that energy stored in glucose and they convert it into another form of chemical energy called ATP. And these two are going to be the basis of the endosymbiont theory. So both organelles are membrane bound and believed to have similar evolutionary origins through endosymbiosis. So our story starts here. We have an old ancient host cell that has perhaps a nucleoid region or a nucleus, and it's bopping around in the oceans, and one day it engulfs a proteobacteria. This proteobacteria, for some reason, somehow gives it energy, and so the cell is therefore better off, and it forms a symbiotic relationship. The bacteria is in a safe space, and the host cell has extra energy. So here we have the host cell with the protobacteria, which eventually becomes the mitochondria. So this is going to eventually become our eukaryotic cell, come billions of years later. And so one of these eukaryotic cells, or prehistoric eukaryotic cells, is going to engulf another bacteria, a cyanobacteria. And similarly to the last story, it's going to give it energy, except it's going to take light energy from the sun and convert it into glucose. And so therefore, it is, once again, better off. The cyanobacteria is better off because it has a safe space, and the host bacteria is better off because it has more energy. And so this becomes our current day plant cell. So what is our evidence for this crazy story? Well, the evidence is that these organelles have a double membrane, just like the nucleus in a eukaryotic cell. Other evidence goes as both of these organelles contain their own set of ribosomes and circular DNA. So both the mitochondria and the chloroplast has its own set of DNA, and it has its own ribosome, so it makes its own proteins to do function in its own being. So it kind of has its own set of DNA, RNA protein. And so finally, our last piece of evidence is that they reproduce independently of the host cell. So not only are they making their own proteins and have their own DNA, they also reproduce independently. They have their own sort of cycle and system. And so for these reasons, we believe that these two organelles were once independent. So let's look at each of these organelles a little bit more closely. Mitochondria are found in nearly all eukaryotic cells. And so once again, function follows structure. If a cell uses a lot of energy, it's going to have a lot of mitochondria. For example, muscle cells have lots and lots of mitochondria because their job is to use energy, to contract. And so here we have our double membrane. Our outer membrane is smooth. We can see that here. And our inner membrane has these folds in it, and these are called cristae. And they go back to the concept of surface area. It allows for more surface area. And so the mitochondria is where cell respiration occurs. And so this increase in surface area allows for a greater number of chemical reactions to occur to make cell respiration more effective, more efficient. And so remember, keeping in mind that these are all different compartments. Compartments allow life to happen. We have the cytosol. We have the intermembrane space that's in between the two membranes. And then we have the inner membrane space. And each of these spaces have a different pH and have different characteristics, which allow for the chemical reactions to occur. And now we have chloroplasts. So chloroplasts are found in plants. The light interacts with a pigment called chlorophyll. Chlorophyll are found in these little stacks. These little stacks are made of pancakes or little flat layers called thylakoids. So the thylakoids are the pancakes, and if you have a whole stack of these pancakes, these are called, it's called a granum. And so we have our outer space, we have our stroma, which is the space between the thylakoids and the inner membrane. The space between the inner and outer membrane is small, and so those are pretty close together. So once again, the concept of compartments is used here. And chloroplasts are closely related to another plant organelle called plastids. And plastids contain either pigment or food. So an example of this would be an amyloplast. It stores amylose, which is a polysaccharide or a carbohydrate. And chromoplast is a plast that is going to store pigments that give fruits and flowers their colors. So it's going to store like carotenoids, so that the carrots are going to have their orange color. Final organelle is going to be peroxisomes. Peroxisomes contain a membrane, a single membrane, and they contain enzymes that can remove hydrogen atoms from substances to help break them down so that they're not toxic, and to turn them into water. So H's are removed, or hydrogens are removed and added to O2 to make water, or H2O. If you remember from our lab in Honors Bio, we used hydrogen peroxide, H2O2, which the peroxisome can remove the hydrogen from it and make it into H2O. And so it is no longer a toxic substance. An example of how peroxisomes form compartments, which is beneficial to the cell, when peroxisomes break down alcohol, they produce the toxic substance of peroxide. Peroxide is toxic to cells, which is why we have to break it down. It is then further broken down into water. But at that point, when it exists in the cell, it can be toxic and harm organelles. But since it's kept separate in this compartment away from organelles, it can't harm its function. So that is 6.5, and at this point I have decided to make 6.6 its own video. So this is just 6.5.